transport. And lastly, we have the bulk transport mechanism. What is this bulk transport? Here, mainly the large molecules will be taking place. The large molecules will be passing through this membrane, and such type of transport is said to be the bulk transport. This will happen in two ways. It can be exocytosis, that is expelling the cargo outside. It is called exocytosis, and sometimes the molecules will be ingested inside. It is said to be endocytosis. So simply in this bulk transport mechanism, we don't have any proteins which are associated. The membranes are slightly modified here. Invaginations are taking place. For exocytosis, the membrane will be, for endocytosis, the membrane will be invaginated, and sometimes the depressions are created on the membrane, and finally they will form a spherical vesicular structures, and through which the functioning will be taking place. Simply in order to make you understand this concept, we will see first regarding the endocytosis. Endocytosis is simply, it is taking place with the immune cells. So WBC cells. So let us say this is the bacterial cell, and this is invaded, and the cell recognizes this bacterial cell with some kind of protein, and it will throw this bacterial cell towards the lipid bilayer. Then quickly, due to this fluidity of the membrane can change its shape. What it will do, it will quickly invaginate, and it will make this bacteria to enter into this lipid layer, and it will seal this membrane like this. Okay, once the bacteria it is kept inside the lipid layer, the lipids are formed here. So all the lipids, they will be forming here, closing or sealing this membrane and capturing this bacteria inside. Now what will happen, this lipid part, this lipid vesicular structure, I have told you that the vesicles are formed by these lipids. So all these lipids, they will form what you can say cage-like structure around this bacterial cell, and this vesicular structure, it is slowly moved and it is joined with the lysosome. So, or you can say WBC cells, we have phagocytosis mechanism. This is simply the phagocytosis mechanism. When there is a bacteria which is invading the cell, quickly the WBC cells will be attacking them, and the bacteria, it is coming like this. So this is vesicle made up of lipids, and inside we have the bacterial cell. This bacterial cell, it is captured inside this vesicle, and this vesicle in turn, it will join with the WBC cell. It is ingested into the WBC cell, or sometimes into the lysosomes, and quickly what it will do, the WBC cell, this is WBC cell, and this is the bacterial vesicle. Okay. Now what will happen? The WBC cell or the lysosome, if it is lysosome, it is suicidal bag. Quickly it will be killing the bacteria by lysing it completely. In the same way, the WBC will also ingest this bacterial cell, and it will kill this bacterial cell. Okay, so this is how the endocytosis will take place. And next, coming to the exocytosis. Exocytosis is movement of molecules from cell inside to cell outside, or you can say from cell interior to the cell exterior. And in this endocytosis, again we have another mechanism. So if a solid substance, it is engulfed inside, or if it is trapped inside, such mechanism is known as phagocytosis. Okay. So endocytosis can be phagocytosis if the substrate molecule is solid. If the liquid is ingested into the cell, so phagocytosis, this is generally associated with the immune cells. Okay, phagocytosis is a kind of endocytosis where the substrate is a solid substance. If the liquid type of substance, if liquids or fluids, if they are entering into the cell cytoplasm through this membrane, we call it as penocytosis. Penocytosis is also called as cell drinking, as it is having this fluids, as it is engulfing, or you can simply say, as it is absorbing the fluid molecules inside the cell along with the fluid. If the solute molecules are absorbed, we call it as penocytosis. The other term is the cell drinking. Cell is drinking something. And phagocytosis is called cell eating. Okay, so what the cell will do? It will eat. Why? Because it is solid substance. We call it as cell eating. And so these are the two types of endocytosis mechanisms. Endocytosis means cell eating. The phagocytosis mainly seen in the immune cells, and it can be cell drinking where the fluid substance, if it is engulfed, we call it as the pinocytosis or cell drinking. This is one kind of mechanism. There is no protein assistance. Simply remember the membrane, it will form vesicles and it will fuse with the different type of substances based upon their need, and they will be acting here. And next, coming to the exocytosis. Exocytosis is called as cell vomiting. Why? Because the substance from cell interior, it will move to the cell exterior. So it will vomit or it will export the substance outside. So the other term which is given for exocytosis is cell vomiting. And in this cell vomiting, I have told you different type of substances can be formed inside the cell cytoplasm. Just say like hormones, we have neurotransmitters. They will be produced inside the cell with the help of organelles, and these substances has to be expelled outside. It has to be released into cell exterior, and this will happen with the bulk transport mechanism, which is said to be exocytosis mechanism. And this exocytosis is also called as cell vomiting. Let us say the Golgi apparatus, it is producing some hormones, and what will happen? This is the hormone which is produced here, and this hormone, it is producing a vesicle around it. The Golgi apparatus will even have the membrane. So the membrane, it will be pinched off. The lipid molecule, it will be pinched off. It will be sealed, and it will form a vesicle like this. And what will happen? This vesicle will come and join with the membrane. So let us say the insulin hormone which is produced inside this vesicle of the Golgi apparatus, it is quickly pinched off from the Golgi apparatus, and it will be fused with this membrane. Okay, so here the insulin which is produced, let us say some insulin hormone or any kind of neurotransmitters that are produced, are captured or sealed inside a bag, that is the vesicle. So this is a vesicle which is pinched off from the Golgi or endoplasmic reticulum, and this is quickly sealed with this membrane to be exported outside the cell. And when this is fused, these are also, the vesicle is also made up of lipids. So lipids, they will be mixed with this membrane, and this membrane in turn, it will be protruded outside and ultimately releasing this vesicle outside the cell. So how it is released? Let us say here is the vesicle, and this vesicle is having the insulin hormone. Let us say this is insulin. Okay, and this insulin, it is released outside the cell. Okay, so this is how the cell will be forming. So this is the lipid bilayer. Okay, this will be the layer which will be present around it. So this will be the lipid bilayer which is pushed outside, which is protruded outside. Why? Because the insulin now, it is forcibly, it will be expelled from the cell interior or the cell cytoplasm. So once it is released outside, the membrane will be sealed back to its original state, and the vesicle, it is released like this. In that way, the insulin or any kind of hormones or proteins, enzymes, different type of substances or cargo present inside the cells, they can be exported out of the cell by this exocytosis. As they are expelling out these substances, we call them as cell vomiting. Okay, so cell vomiting, cell drinking, cell eating. Cell eating is called as phagocytosis, mainly seen in the immune cells, the WBC cells, and cell drinking, that is the pinocytosis, if the endocytosis or entry, or you can say the inward movement of these large molecules, if they are taking place. If it is solid, we call it as cell eating or phagocytosis. If it is liquid, we call it as cell drinking, the pinocytosis. And if it is expelling the substance outside from the cell interior, we call it as exocytosis. Exocytosis can also be called as cell vomiting. Okay, so this is regarding the bulk transport mechanism, where lot of energy, ATP, is required, highly expensive process even compared to that of the active transport. Okay, so this is regarding the transport mechanism on the membrane, the plasma membrane, and for you I will be giving a quick revision of the details of the transport mechanism on the membrane once again. So this is overall view of the transport, the classification of the transport which we have discussed in detail, children. Without leaving any point, I have written all the things which we have discussed. So this is the thing, the transport. So we have the three major classifications here. The transport is divided into passive transport, active transport, and in the center I have given the bulk transport. Okay. So first we will see with the passive transport. In passive transport we have discussed with the simple diffusion. There is no necessity of the protein here. Proteins are not required. If the protein it is required, we call it as facilitated. Both are diffusion. Essentially the passive transport, I have told you, it is the osmosis. Okay, so here the osmosis is taking place, the diffusion of molecules is taking place. It is taking place from higher concentration to the lower concentration along the concentration gradient, which is a basic potential for the molecules to move in and outside the cells. Okay, there is no requirement of additional energy like ATP or other molecule support. It is not required here. So in this case, the passive transport is further divided into simple diffusion and facilitated diffusion. And another important common point for this passive transport, it is the movement of single molecules. Always a single molecule is participated, and we call these systems as uniporters. Why? Because it is not for simple diffusion. Protein is not present here. The protein, it is present only for the facilitated diffusion. The movement of a single molecule with the help of a protein, we call it as uniporter. So all these categories coming under the facilitated diffusion, they will be called as the uniporters. Okay, the enzymes or the proteins essential for their transport are uniporters. Now let us see. Simple diffusion, it is mainly taking place with the non-polar small uncharged molecules. Even the gaseous molecules, they will be having the easy entry into the cells without the necessity of a protein on the membrane. Simply through the membrane pores, there are certain regions which are known as porosomes. I have told you, loosely associated lipid molecules. Through these regions mainly, or through the gaps that are present in these lipid molecules, these molecules will be easily passing. Okay, certain type of hormones which are non-polar are also being passed here. And coming to the facilitated diffusion. In facilitated diffusion, here if you observe, the entry of the molecule, it is through the protein. So except the simple diffusion, everything which we are discussing is nothing but facilitated with the protein here. But facilitated diffusion is a term given for the passive diffusion. Okay, so facilitated diffusion, the uniporter system, let us see. This is again divided into two categories I have given. What are the two categories? About the proteins associated in the facilitated diffusion. So here we have two proteins, children. One is channel protein creating pore, and other is a carrier protein which is making the conformational changes. It will be changing their shapes gradually or rapidly with the signaling of the molecules. And in this, the channel proteins will create a pore, and again they may be open always. We call them as non-gated. Examples of the aquaporins for water channels we have discussed, and we have gated channels. They are always closed, and temporarily they will be opened with a signal. So opening and closing, the shutting of the valves, it is highly controlled by the signals mainly associated with these gates. And what type of signals? Based upon the signals, again we have discussed we have categorized these gated channels as two important categories. One are the ion gated, another is a chemical gated. We even have the other two gated channels like mechanical gated, ligand gated, that we have not discussed here, and it is not essential to be described all these things here. So mainly we have the ion gated channels and the chemical gated channel. Ion gated channels are mainly associated, or you can say they're activated with the ions like magnesium, calcium, or you can say potassium, chloride. All these ions, they will be activating these ion channels, and based upon their regulation and their charge on the membrane, mainly these channels are opened and closed. Okay. And coming to the chemical gated channels, chemicals like neurotransmitter and hormones mainly playing the essential role in the controlling the chemical gated channels. Okay. This is regarding the gated and the non-gated channel. Coming to the carrier proteins are highly specific. These are non-specific. Channel proteins are non-specific because different type of substances they will be having entry when the channels are open, like pores. Okay. Carrier proteins are highly specific. They will not create pores. Simply, they will be changing their conformation to allow the molecules in and outside the cell. And the example which we have discussed regarding the amino acids, why I have not given any name for the particular amino acid, because generally a single amino acid, it is not entering into the cell, children. Amino acids are always taken inside the cell or imported into the cell in a group, and hence I have given the example of amino acids here. So amino acids, they will be attached to a specific site of attachment present on the carrier protein, and this is the one of the example of the carrier protein unipolar system under facilitated diffusion. Coming to the active transport, first we will discuss, later we'll be discussing with the bulk transport in sequence. Okay, so next we have discussed with the active transport. In active transport, mainly we have three categories. One is primary active transport, secondary active transport, and group translocation. In primary active transport, what type of proteins are present? Okay, so that we will see first. In primary active transport system, you have to remember the active transport, it is mainly uphill movement, it is against the concentration gradient, and it is called as the reverse osmosis. Okay, and the proteins which are playing mainly which are present in the active transport are all our carrier proteins. They will show the conformational changes which are mainly seen in the case of facilitated diffusion. The same proteins will be present, but based upon or their differences in their functioning, they are slightly modified with their names that we will see now. So in primary active transport system, the first classification under the active transport, primary active transport, where ATP is really required here for the operation of these carrier protein molecules. And as these carrier proteins are operated in the presence of ATP, they are pumping the molecules against the gradient with the excessive additional energy that is from the cellular energy is utilized. We call them as pumps or ATPases. Why? Because the specific site of attachment, even you have to remember the ATPase is mainly the enzyme which is associated with the hydrolysis of ATP, and hence we can call these carrier proteins as pumps or ATPases. And here we have two type of systems. One is the unipolar system. Single molecule can be passed inside and outside the cell, or more than one molecule can also be transported in and out of the cell. If single molecule is transported to the carrier protein, we call it as a unipolar. If it is more than one molecule, we call them as the co-transporters. The co-transporters, again I have told you, if they are moving in the same direction, they are symporters. If they are moving in the opposite direction, we call them as the antiporter system. And here I have given examples, some of the examples for primary active transport. Sodium-potassium pump, which is the most essential pump to maintain the sodium gradient inside the cell cytoplasm and the cell exterior that we have discussed before. And another examples which I have given here, the hydrogen ion pumps and calcium ion pumps, magnesium ion pumps, also known as magnesium ATPases, calcium ATPases. You can call it as ATPases or pumps, any way you can call them. All are these carrier proteins showing conformational changes, but they are associated with the ATP hydrolysis for their working. And coming to the secondary active transport, here ATP is not required directly, but energy is essentially present here. So for the movement of molecules, the innate potential, it is not used or not present for one molecule. Mainly the more than one molecule is participated in the secondary active transport, and I have told you that this is a kind of obligate relationship where each molecule will be supported. Any one of the molecule in the couple system will be supported by the other molecule. And how it is supported? One of the molecule, it is moving downhill, another molecule will show the uphill movement. The uphill movement requires extra energy, and this extra energy, it is sourced not from the cellular currency, that is ATP, but from the companion molecule which is always present with it. Okay, so always the secondary active transport, it is guided with the co-transporter systems. Essentially or exclusively, you can see the co-transporter systems operating in the secondary active transport. So these co-transporters again can be symporters or antiporters based upon the direction. And here I have given some examples, children. Sodium, glucose, Co-transporter, sodium chloride co-transporter. These are two examples which I have discussed in the previous videos. And we have the sodium potassium chloride. These are also present on the kidney cells. And we have sodium iodide, sodium and iodide channels. These are mainly related to the thyroid gland and mammary glands in order to absorb the iodine into the milk. Mainly in the mammary glands it is required. And also for the thyroid functioning, this receptors, or you can say the carrier proteins, are present on the thyroid glands. And next, coming to the sodium calcium, this is for maintaining the calcium equilibrium, or you can say the amount of calcium inside the cells for bone development and all. You can see the sodium and calcium. If you observe, mainly all these gradients are associated with the sodium. This will make you understand how the sodium is important inside our body to maintain these gradients. So without the sodium gradient in the extracellular region, the many molecules will be stored without the uptake, without any intake into the cell cytoplasm. Mainly it is acting like a helper molecules inside the cells. And in this again we have different examples I have given. One is importer and antiporter. So these two terms are related to the co-transporters of primary active transport as well as the secondary active transport. If you want, you can draw a connecting line so that it is easy for you. So co-transporters for both the primary and secondary active transports, they are again divided into symport having same direction, antiport if they have the opposite direction. And lastly, we have discussed with the bulk transport. Here it is not facilitated with any protein, any kind of protein, it is absent. For the recognition of the molecules, we have proteins. Children, don't get confused here. The movement of molecule, it is mainly taking place with the help of the membranes by formation of the vesicles we have discussed. Why? Because large amount of substances will be moved in and outside the cells, and all this movement, it is mainly supported with the formation of the vesicles, micelles we have discussed. And in the system, mainly the molecules which are approaching the cell, which are docking the cell, they are mainly signaled, or you can say identified by the receptor molecules. They will be helping in identification, but passage of these molecules will be done with the help of the vesicular movement. Okay. So in this bulk transport, we have two types again: exocytosis, cell vomiting, that is expulsion of substances, and endocytosis, that is admission of substances into the cell. Endocytosis, again, it's solid substrate, it is engulfed, or you can say it is taken inside the cell. We call it a cell eating or phagocytosis, mainly seen in the immune cells we have discussed with an example. And next we have the pinocytosis, the cell drinking. Okay, mainly the fluid substances, any type of substances in the fluid nature, if it is absorbed or left out of the cell, we call it as a pinocytosis. Okay, so this is also called as cell drinking. Okay, so this is regarding your bulk transport. So overall, if you observe with the transport, we have discussed all these concepts. Children, it requires a lot of practice and lot of revision, and even you have to memorize many terms. Mainly regarding the transport system, you have to concentrate. For NEET, mainly you have to concentrate on the passive transport, simple and facilitated diffusion along with examples, and active transport, primary active transport, and secondary active transport. Other than this, we have an extra information regarding transport for group translocation and bulk transport, which is not included in your syllabus, or you can say for your NEET. For other exams, it is very essential. So mainly the NEET aspirants, they can be concentrating on the passive as well as the active transport. Okay, so this is regarding your lecture today. We'll be meeting in the next lecture with the discussing with the plasma membrane functions, other functions which we are left out with the plasma membrane. Thank you.